I'm Elizabeth Bowen, conference producer at Technology Networks, and I'm here to introduce our next talk, Improved Sample Preparation, Separations, and Data Acquisition for Single-Cell Proteomics. I'm pleased to welcome Dr. Ryan Kelly from Brigham Young University. Dr. Kelly specializes in pioneering ultrasensitive biochemical analysis technology. After earning his PhD in analytical chemistry from BYU in 2005, he returned to BYU following a 13-year stint as a staff scientist at the Pacific Northwest National Laboratory. Dr. Kelly's recent work concentrates on single-cell and spatial proteomics, where he refines sample preparation, separations, ionization, and mass spectrometry for low-input samples. His team's efforts have facilitated rapid profiling of thousands of proteins from single cells. A warm welcome to you, Dr. Kelly. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk. So, feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Dr. Kelly's presentation. You can also benefit from some fantastic supporting literature, which you can download for free below the video player. If you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. I will now hand over to Dr. Kelly to begin his presentation.
Okay, thank you very much. I'm excited to share some of our recent progress in the field of single-cell proteomics. So to sort of motivate the reason why we're interested in analyzing single cells, I have kind of an analogy of taking a complex electronic device. In this case, this is an Amazon Echo smart speaker. And if we wanted to study that smart speaker, imagine if the first thing that we did with that was take it, put it into a blender and produce this mesh of smart speaker dust. You can see that would give us plenty of material to analyze, but we would be measuring, even if we could perform an accurate chemical analysis of this dust, we would be measuring an average of the constituents from that smart speaker, and we would lose information regarding composition of trace components that might get averaged out and fall below our detection limit, and we would lose all information related to spatial organization. So it would be quite difficult, even with a perfect chemical measurement, to go from this to reverse engineering a smart speaker. Biological systems are much more complicated than that. And so the motivation for single cell analysis is to essentially dissect the tissue and measure each cell individually for a more nuanced understanding of the biological system. And this would allow us to track the original spatial organization. We can identify trace components because they're not kind of being blended into the background, but it poses some challenges in terms of measurement sensitivity, because now each sample is much smaller than the original sample, and challenges in measurement throughput, or the number of samples we can measure per unit time, because we've just made a lot more samples that need to be measured. So we looked at this field of proteomics that had been limited to bulk scale samples comprising thousands or millions of cells. And we recognized that the instruments had become quite a bit more sensitive over the years, but that sample preparation was, we reasoned, quite limiting for small samples. You can see if we take a conventional sample through a multi-step sample processing workflow, lysing the cells, pulling out the proteins, performing a number of chemical and enzymatic steps on that, cleaning it up. If we start with a large amount of sample, that's no problem. There are these surfaces, proteins are sticky, so we will lose some of those. And there are all these transfer steps, so we will kind of multiply those losses. But if we start with plenty of material, we'll have plenty of material to measure at the end. But if we start with a small sample, such as a single mammalian cell, we might lose everything going through this process. So we hypothesize that a more appropriate sample preparation workflow for single cells would be beneficial. We also, the second challenge with working in large volumes for single cells is that if you lyse a cell in a volume of 100 microliters, you're essentially diluting the contents of that cell by a factor of 100 million. And so since protein digestion rates are proportional to the concentration of the proteins being digested in the sample, at least at low concentrations, it's like taking a grape and squishing it into a large swimming pool is the amount of dilution. So starting with low protein concentrations that we want to digest should lead to poor digestion efficiency. So to reduce our volumes and our surface contact areas, I had this great partner in crime that joined my lab, Ying Zhu. He joined from a research group in China where he had been developing robotic nanopipetting platforms. So you can imagine this is a robot that can pick up and dispense nanoliter volumes of liquid. And rather than going into a large vial, we just engineered or we microfabricated a glass chip, glass microscope slide that had these hydrophilic spots surrounded by hydrophobic background. And so that limited our volumes by nanopipetting, that limited the volumes that we exposed our samples to, and it reduced the amount of dilution because now we could work in low nanoliter volumes, a couple hundred nanoliters. So we called this workflow that resulted from this nanoPOTS, which stands for Nanodroplet Processing in one pot for trace samples. And you can see we're only ever adding reagents or getting a sample into one of these nanowells, and then we would add reagents, incubate, add the next reagent, incubate. So everything just stayed in this one vial. And then that allowed us to, or in this one nanowell, and that allowed us to minimize dilution and minimize the transfer-related sample losses. And we were able to reduce our sample input initially from other platforms had been able to quantify about 2,600 proteins from samples comprising 5,000 cells. And we were able to reduce our sample input originally to 10 cells and actually increase proteome coverage to more than 3,000 proteins for these 10 cell samples. So we reduced our sample input by a factor of 500 and increased our proteome coverage. So that really showed that sample preparation had been the bottleneck, and addressing those limitations made a large improvement. And the label-free quantification, the run-to-run reproducibility was also excellent with this platform owing to the ultra-precise dispensing. So we were able to move forward with this platform. Our next step was to extend this down to single cells. And so we needed a way to interface and sort single cells into nanowells. So we did this with fluorescence activated cell sorting, where we could just program if we wanted 1 or 10 or 20 or however many cells into a well, we could do that with this laser or with this fluorescence activated cell sorting platform. The proteome coverage was more limited when we dropped from 10 cells to 1 cell. You could see based on MS2 identifications alone, we were getting about 200 proteins per cell, and with match between runs, we could, which is based on MS1 level identification, including retention time as a discriminating factor, we could quantify about 700 proteins per cell with this platform. We could also begin to look at things spatially by combining nanoPOTS platform with laser microdissection. So laser microdissection allows us to take a histological thin section of tissue, identify regions of interest, and pull those into our nanoPOTS platform for analysis. So now we have a way to do spatial proteomics and single cell proteomics. We can also combine both by using laser microdissection to isolate individual cells. And that's what we're doing here in this nice application in collaboration with some great researchers, Ed and Amanda at Biogen, where we're looking at motor neurons that are isolated from spinal tissue sourced from donors, human donors that had ALS, and those that were kind of control donors that were age matched but did not have the ALS disease. This is amyotrophic lateral sclerosis, also known as Lou Gehrig's disease. It's a disease associated with degeneration of motor neurons in the spinal column. And so here we could isolate, from spinal tissue, we could identify individual motor neurons, excise those from the tissue using laser microdissection, and analyze those with the nanoPOTS platform. We could also kind of follow the disease, at least we hypothesized that we could, by finding motor neurons in the ALS tissues that resembled wild type or control pathology. So this is staining for a single protein, TDP43, that forms these crazy aggregates in the tissue. So a control motor neuron, this is how TDP43 stains within that. And in an ALS donor tissue, we could also identify similar looking TDP43, and then we could see the TDP43 start to accumulate and form these aggregates, so we could sort of stratify the motor neurons within an ALS donor of resembling no pathology in terms of TDP43, mild, moderate, or severe, and we could follow this disease and look at how other proteins are changing their protein expression, going from resembling control to a more severe. Some kind of drop and start to come back up, things like that. So we think this is a nice way to study these neurodegenerative diseases that combine single cell proteomics and spatial proteomics. We can also just do a straight up spatial profiling. This is in collaboration with my colleagues at Pacific Northwest National Laboratory. We started this before I moved to Brigham Young University. This was taking a mouse tissue, a uterine tissue prior to embryo implantation, and So this is the luminal epithelium region that's highlighted here, surrounded by stromal tissue. And we could essentially form pixels, microdissecting each one of these, transferring it to the nanoPOTS platform, and then we can look at which proteins are being downregulated in the luminal epithelial tissue relative to the surrounding stroma, or upregulated in the luminal epithelium. And we could do this for about 2,000 different proteins. So this is just showing 12 here, but we could perform kind of query 2,000 different proteins and see how they're changing. So from this initial work with nanoPOTS, also some great work from Nikolai Slavov out of Northeastern University, collaborating with Bogdan Budnik to develop the SCOPE MS workflow for multiplex proteomics. This really got the community, the NIH, and the broader research community more interested, to where we started seeing calls specifically for single cell proteome analysis. This is on the sort of nonprofit or academic side. We also saw some small business calls from the National Institutes of Health here in the US requesting technology development for single cell proteomics. And the instrument developers also took notice. You can see some advertising material from Thermo here, talking about a singular focus, one cell, one shared mission. And this is really encouraging for us, because as we researchers work hand in hand with instrument developers, it really moves the whole workflow forward. And it was refreshing to see the vendors jump on board, because when we would run into trouble in the early days in 2017, 2018, we said, we can't see anything from a single cell on, say, our Orbitrap instrument. They would say, well, you're not really supposed to. They weren't designed for that. But since then, they've really embraced and developed new platforms for single cell proteome profiling. Bruker has also launched an instrument specifically devoted towards single cell proteomics. That's what the SCP stands for. More recently, they've developed the Ultra that we're having just installed in our lab, and we're excited to get started with that. So from this original nanoPOTS platform, we've been focusing on increasing sensitivity, measurement throughput, and accessibility to other laboratories, because not everybody could build a robotic nanopipetter and these nanowell glass chips. So I'll start here by talking about some recent efforts in increasing sensitivity. So the reason we care about increasing sensitivity is because we can't, unlike DNA and RNA, we cannot amplify proteins above a detection limit. So we need to bring the detection limit down if we want to study trace proteins. And since global proteome profiling detects proteins roughly in their order of ranked abundance from high to low, so if we can identify 1,000 proteins per cell, we're going to identify the 1,000 most abundant proteins of that cell. And as we drop it down, or as we increase the number of proteins we're able to analyze, we can look at new classes of proteins. So as we go from high abundance to low abundance proteins, we can dig deeper from, say, more structural proteins to more signaling proteins and things like that on the low end. So that's kind of the motivation for increasing our sensitivity and the number of proteins that we can profile per cell. And to do this, we also, one of the ways that we accomplish this is to reduce our flow rates of our so after separation, or after sample preparation, we perform a chemical separation. using liquid chromatography. And we do this at as low of a flow rate as possible, because this is a measure from some experiments that Suchi performed in our lab with direct infusion, going from 1,000 nanoliters per minute or a microliter per minute down to 1 nanoliter per minute with these fancy electrospray emitters that we make in our lab. And you can see as we reduce our flow rates, we increase our ionization efficiency and our ion transmission efficiency into the mass spectrometer. And so as we go from around 300 nanoliters per minute, where most proteomic separations are performed, down to 20 nanoliters per minute, we get about 10 times more signal per analyte molecule. So we've been pursuing these lower flow separations for this very reason, going from 75 micrometer inner diameter columns that operate at about 300 nanoliters per minute, down to 30 micrometer internal diameter columns that operate at 50 nanoliters per minute. And you can see from the same size sample, you get a lot more signal in the chromatogram, reducing your flow rate. And then we've gone from 30 down to 20, and we get an additional gains in sensitivity by doing that. So with a 20-minute analysis using a single cell sized sample at with a 30 centimeter long, 20 micrometer internal diameter column, using standard data dependent acquisition on a Orbitrap Exploris 480 instrument, you can see this is kind of what we can now see using these low flow separations, about 6,400 peptides per single cell sized sample, and about 1,500 proteins per cell using this. And so to do this, we need to make our own columns because there aren't a whole lot of commercial options available. And then for to further increase our sensitivity, we're looking at alternative data acquisition methodologies. So, so this is sort of a specific to Orbitrap instrumentation, but for single cells, longer injection times are required to give us an adequate ion population. That results in fewer MS2 spectra. So it takes longer to acquire each one. So we get fewer of them. But since we're having these long MS2 cycles to fragment ions and measure the fragment ions, if we have these long cycle times anyway, we might as well run the Orbitrap in a high resolution mode. Otherwise, it would just be sitting around in between scans. So it gives us room to resolve multiple peptides in each MS2 spectrum. And so as we were kind of working through these acquisition modes that could take advantage of this, Thermo, in collaboration with MSAID, a company in Europe, developed a new algorithm called the Chimeris search engine that that allows us to identify more peptides from a single MS2 analysis. So that means we can increase the number of, or the window of peptides that we let into the instrument, fragment those all together, and identify multiple peptides in a single MS2 analysis. So we call this wide window acquisition. You can think of standard data dependent acquisition, where we're isolating with a narrow window single peptides to be fragmented and analyzed, whereas data independent acquisition has a much wider window that's co-isolated, and you just simply kind of blindly step through the range of precursor mass to charge values. With wide window acquisition, it's doing data-dependent acquisition, but with data-independent acquisition sized windows. So that means as we isolate this ion to be fragmented and analyzed, we widen our windows so we also pick up some neighboring peptides. And it works. You can see we're going from fast, short injection times with low resolution here over to long injection times with high MS2 resolution over here. And then within each of these, we're just increasing the window size that we acquire from 2 mass to charge units or Thompsons out to 48. And you can see as we do this, as we go slower, we're getting fewer and fewer MS2 spectra. These are the colors in orange. But as we open our windows wider, we get more peptide spectrum matches per MS2 spectrum. So we have, in this case, about 30,000 peptide spectrum matches where we only acquired 15,000 MS2 spectra. So it shows that the Chimeras algorithm is working. And then if we look at unique peptides, so not peptide spectrum matches, but the unique peptides that we get, you can see within each of these resolution and injection time combinations, we get sort of a maximum where we're co-fragmenting and analyzing multiple peptides at once. And then if we go wider and wider, it looks like we start to struggle and are not as effective. Maybe the spectra are becoming too complex. So there's a sweet spot between about 8 and 18 Thompsons that we can work at. And in terms of, so this is peptide coverage for single cell sized aliquots of a human protein lysate digest. And this is looking at the protein level. With MS2 based identification alone, now we're able to quantify more than 2,000 proteins using the low flow separations and the wide window acquisition. And then if we include also the match between runs based identifications with a 40-minute analysis, we get to about 3,600 proteins per sample. And we can also speed this up and make our chromatography go twice as fast, doubling our throughput. And as we do this, we only drop, 10 to 15% of our proteome coverage. So that's a worthwhile trade-off for many applications where we would like to go faster. And in terms of differentiating different cell types, that becomes very, very easy with this wide window acquisition. And here we're looking at actual single cells. So our proteome coverage has dropped a little bit as we go from single cell sized aliquots to true single cells. Now we're getting about 3,000 proteins per cell. And that's because we're still probably losing something during sample preparation. But you can see if we compare data-dependent acquisition to data-dependent acquisition without match between runs and with match between runs, and our new wide window acquisition, we get the greatest proteome coverage with the wide window acquisition, especially if we're comparing MS2 based identifications, which are a little bit more confident. We can still, easily separate in a PCA plot K562 cells from HeLa cells. This here is a histogram of how many proteins are expressed at a given copy number per cell based on a bulk analysis. And so single cell proteomics is biased towards the most abundant. This is kind of our previous work with single cell proteomics. Our most sensitive workflow before we did this wide window acquisition. And with the wide window acquisition, we're able to look at proteins expressed more in this 10,000 cells to 100,000, or copies per cell to 100,000 copies per cell that we previously missed. So it allows us to dig about an order of magnitude deeper into the proteome. I'm going to skip over this in interest of time, but we can look at single gene knockouts now. Here we knocked out a single gene that expresses a protein related to autophagy, and we can look at how that kind of perturbs other proteins within the cell. Still, we're getting about 3,000 proteins per cell in the control cells and the knockout cells. We're still, even though we only knocked out one gene, we're still able to tease apart these, and we can find about a couple hundred proteins that are significantly up and down regulated, kind of relating to this autophagy pathway. So a single perturbation, and then looking at how hundreds of proteins are up and down regulated as a result of that. We're also interested in increasing measurement throughput. And so one of the ways we can do this is through multiplexing, like the Slavov and Budnik's SCOPE MS protocol. Well, we said that only can get up to 18 plex right now. So we could also kind of combine multiple layers of multiplexing on top of each other. In this case, this is non-isobaric or isotopic labeling with heavy isotopes using stable isotope labeling in cell culture or SILAC, plus tandem mass tag isobaric labeling to get from 18 plex, but you always lose a few due to boost and carrier channels. So we did this multi-step or multiplexed, or what we call hyperplexing, not our original term, but hyperplexing to get higher order multiplexing here. All right, so this bottom line, this allows us to quantify about 300 proteins per day using short gradients, or 300 cells per day, or 200 cells per day with longer LC gradients that give us a little bit greater proteome coverage. And looking at 4 different cell types, we can tease those apart, and you can see that the heavy and light versions of a given cell type line up right on top of each other, but the 4 different cells are easily differentiated using this approach. We can also increase our measurement throughput by avoiding the dead time that is inherent in liquid chromatography. So the part we care about from liquid chromatography is when peptides are actively eluting. But then we need to wash the column, load a new sample, regenerate the column, then there's a gradient delay, and then the peptides start eluting again. So as we go to low flow separations, the time during which peptides actively elute becomes smaller and smaller relative to these overhead steps. So we reason that to have fast separations that are also high throughput, and where we're not having the mass spectrometer sit idle for a lot of the time, we can develop these multi-column systems. And I don't really have time to go into the details here, but this geometry where we elute samples into a storage loop first allows us to elute one sample into the mass spectrometer while another sample is being transferred into a storage loop while we're regenerating the other column and getting it ready for the next separation. And at the same time we're doing all of that, we can be auto-sampling the next sample. So we kind of, instead of having one sample go through the liquid chromatography system at a time, we can have multiple samples being making their way through the system. And you can see this is a 2-column system where we're going from one column to the next and back to the first column. So column 1, 2, 1, 2, 1, 2, and the chromatograms look roughly the same. The proteome coverage for each one of those also looks roughly the same. And then we've said, well, okay, that's good for maybe 30-minute analyses, or 48 samples per day. Can we go faster? We want to have separations that are fast, still have high peak capacities, low flows, and 100% duty cycles. And so that has been our challenge, and we're developing improved separations. This one has a peak capacity of about 100. That's typically what you'd see for an hour-long separation, but we're doing this in 5 minutes, and still it's only 20, our flow rate's only 20 nanoliters per minute. So we think that's one thing we can do. Of course, we haven't multiplexed this into a multi-column system yet, but once we do, then that would allow us to analyze about 288 cells per day in a label-free workflow. So the last thing I'll talk about is accessibility. We want others to be able to do these analyses as well. And so, as I showed before, the nanoPOTS workflow was a multi-step workflow, 1 pot, multiple dispensing and incubation steps, and it took about 1 day for sample prep. Our goal is to have 1 pot, but just 1 dispensing step, have the whole thing achieved in 1 hour, and achieve the same proteome coverage. So we've explored combining the steps from the multi-step workflow into a single step, looking at what do we keep and what do we omit. Do we keep this reducing agent, DTT, or do we throw it out? We got rid of alkylation in this, and you can see if we get rid of the reducing agent, but have just the protease or the protease plus this non-ionic surfactant, we get pretty good proteome coverage doing this. And this is taking advantage of a new protease released by the company Promega. So you can buy this Promega rapid trypsin lyse C mixture, and it's stable at 70 degrees C. So we can do the digestion at 70 degrees C. That enhances the kinetics, and it's the same temperature that we were previously using for cell lysis before we did a 37 degrees C digestion. So this allows us to do everything at 30 C in 1 hour, and the result is that incubating single cells in this rapid trypsin lyse at 1 hour with or without the non-ionic surfactant dodecyl multicide works well for label-free single-cell proteomics. And we get about the same proteome coverage from the multi-step workflow as we had for the 1-step workflow. The peptide hydrophobicities were similar, peptide intensities were similar. And then we partnered up with Hewlett or HP Inc. They're using their inkjet printing technology that you're probably familiar with to dispense single cells into well plates, and then dispense single nanoliters of reagents into those well plates. So this is a low-cost robot for cell and reagent dispensing that we can now use for single-cell proteomics. We first looked at the volumetric dispensing capabilities here, found that that was good. Sorry, and then but it's really made to work with well plates, 384 well plates, not nano wells. But we reasoned that if we limit the volume in a PCR plate, the bottom of that PCR of the well in a 384 well PCR plate is similar to that of a nano well. So we can, using this workflow, using the wide window acquisition, we can get almost as many proteins as we had previously with that nanoPOTS workflow. All right, so so this is an excellent, and also this HPD100 is now being marketed as the TCAN Uno. They've partnered up with TCAN, but it's an excellent no compromise solution that's very accessible, because a lot of people can work with well plates. And this is actually now published. I should have updated that. And it gives us flexibility to do this in essentially any proteomics lab. So I'm just going to wrap up here. We've seen a lot of advances in sample preparation, separations, mass spectrometry, acquisition, and data analysis. Single-cell proteomics is becoming increasingly robust, quantitative, and easy to perform. Hyperplexing can get us to about 300 cells per day currently, and wide window data-dependent acquisition gives us a proteome coverage of 3,000 proteins per cell. This time, I'd just like to thank my great research group here at Brigham Young University, and thank the National Institutes of Health for sponsoring our work. And I'll be happy to answer any questions at this time.